Hello, my name is Jewel Mitchell, and this is my exam mini video number 6 for Genetics 311. Today I'm going to talk about developmental networks, and we're going to look at a specific example that leads to the formation of spikes or no spikes in certain cells of the skin of a thorny dragon. So first, what is developmental network? A developmental network is a pattern in DNA that determines whether or not a specific gene will be expressed due to the presence or absence of proteins on the strand. So here we have our example. S is going to represent UVA, P is going to represent Duke, I is going to represent NC State, K is going to represent UNC, and E is going to represent Wake Forest. And this either spikes or no spikes is going to be who wins or loses the ACC National Football Championship this year. And so the arrows represent the activators, and these T's represent inhibitors or repressors. So for our wild type, UVA would lose and would activate Duke, and then Duke would win, which would inhibit State. State would win, which would inhibit UNC. UNC would lose, which activates Wake Forest, and Wake Forest will win, winning the ACC championship. So if there's a mutation with Duke and something happens and they're out of the tournament, then State would beat UNC, and then UNC would not be able to play against Wake Forest, so Wake Forest would not be able to move on in the ACC championship. So now NC State is out because of a mutation, so UNC would play Wake Forest, and Wake Forest would then win the championship. So if NC State and Wake Forest are both out because of mutation, UNC would not get in their last game, so they would not go to the ACC championship. So if UVA and UNC are both out because of a mutation, UVA would not even be able to play Duke in the first round, so then Duke cannot inhibit NC State anymore, and NC State would not play UNC anymore because they are out, so they would miss that game, and then Wake Forest would never be activated, so they would never go to the championship. So going back to spikes, one type of regulation that could explain why not all cells in the skin have spikes, even though they have the exact same genome sequences, is DNA methylation, which refers to the addition of methyl groups to the nucleotide bases. So, many epigenetic effects are caused by changes in gene expression that result from alterations in chromatin structure or other aspects of DNA structure, such as DNA methylation. It is often used as an epigenetic signaling tool that can fix genes in the off position.